Hello, everyone. Camille and I, Mary Power, would like to welcome you to this next really exciting Lectures in Retirement series. And it's a very great pleasure today to welcome my friend and colleague, Britt Koskella. Britt is a professor in integrative biology at Berkeley, and she does absolutely fascinating research on the evolutionary ecology of three, what you'd say are levels in food chains. The bacteriophage that eat bacteria which may be the enemies of plants or snails or us, and the hosts, plants or snails or us. So she's done really fascinating work that over all sorts of temporal and spatial scales, time with coevolutionary races, and spatial, for example, with plants from stomates to leaves to different branches in a tree to different trees in a forest. So she really can think very fluidly about these interactions at both evolutionary and ecological scales. And it's wonderful to welcome her as a speaker in this series. And her talk today will be the broad-reaching impacts of bacteriophage viruses from bacterial evolution to microbiomes and human health. Thanks, Britt.
Wonderful. Thank you. Okay, let me share my screen here. It's been a long time since I gave a talk on Zoom. I still have lots of Zoom meetings, so this is bringing me back to the start of lockdown. And just like the start of lockdown, I have my dog here in the office. So we'll see. It's a 50-50 chance whether there's going to be what used to happen during the beginning of the pandemic all the time, a dog interrupted talk, but we'll see. I'm really excited to be here. Thank you so much for the invitation. Mary was indeed the chair of the search committee on which I was hired, and I'm grateful every day because I absolutely love being here at Berkeley and engaging in this research community. And one of the reasons is because of this broad interest and the fact that people are keen to hear about work being done outside of their field. So I am very happy for folks to interrupt at any stage here. So if you have questions, of course, you can use the chat or you can wait until the end. I will make sure I have time at the end, but please do feel free to just either raise your hand or speak out, especially if they're clarifying questions. And then my only other disclaimer is I do have a lot of data figures on the slides, and I will always try to present the sort of at two levels. But if I'm not going into detail enough for you about, how the data was generated or what the specific analyses are, of course, please reach out or you just raise your hand and ask. I'm happy to go into more detail. But what I really wanted to do today is go talk through where we are in terms of our understanding of bacteriophages. So bacteriophages, as I'll describe, have been they were discovered before antibiotics. So we've known about viruses of bacteria for some time, but they're really coming into their own right now. And some of that is for good reason. It's just, there's finally tools, viromic tools that didn't exist even last year or the year before. So we are rapidly uncovering this huge amount of viral diversity that we did not know about, but some of it is for not so good reasons, because we're in the middle of or on the precipice of a true antibiotic crisis. And suddenly the need for alternatives to antibiotics are becoming urgent and clear. So what I wanted to do today is talk about what we know about how bacteriophages shape bacterial cell phenotype and function, as well as bacterial competition, how we can use that information to create phage therapeutics, but then broaden it out a little bit in the second half of the talk, because one of my passions at the moment is moving beyond the idea that you have a phage targeting a specific bacterium and start to think about how phages are playing a role in the broader picture of the microbiome. So many of you in the audience will know that the microbiome is also in its heyday at the moment, as suddenly we're realizing a critical part of our health, especially our gut health, but as our lung health, our skin health, our nasopharynx, defenses against disease. And it turns out phages play an important role there as well. And then at the end, if there's time, I'll sort of speak a little bit to what we don't yet know, which is a lot about how phages are shaping these interactions and eukaryotic host phenotype. Okay, so that's the goal. We'll see how much we get through. But just to make sure we're all starting on exactly the same page. When I'm saying phages, I'm primarily going to be talking today about lytic phages, which you see here on the left. And these are phages that bind to and inject their DNA or RNA into a bacterial cell, hijack the cell replication machinery, produce new viral particles, and then lyse the cell open in order to transmit to future cells. That's what we refer to as lytic phage. But B and C here, these panels in the middle and on the right, are two other lifestyles of phage. On the far right is filamentous phages, and these are very interesting and highly understudied, and I actually won't talk about them at all today, unfortunately, for time reasons. But these are phages that replicate within the cell and sort of ooze out, but do not actually kill or lyse their cells. In the middle here are the temperate phages, and these are phages that are capable of being free viral particles, as you can see at the top, and can indeed infect cells. But then there's a sort of decision-making process that we're learning a lot about how that decision is made based on environmental information. But these bacteriophages are capable of integrating into the bacterial genome and then replicating along with their bacterial cell host for multiple generations. And if time let's say if the environment becomes stressful or they have other information that it's a good time to get back into the free viral form, they can actually jump out of the bacterial chromosome, start replicating and enter the lytic cycle, lyse the cells, and start the whole process again. And like I said, I won't speak much about these temperate phages. Unfortunately, I know there's a lot of fans in the audience, but I will at least say that these temperate phages play a critical role in shaping what bacteria can do. And what I mean by that is this is a work not from my lab, but from another lab, and it's just one example of many, where if you look at this one bacterium, Actinobacteria baumannii, you can see that on this phylogeny that if you start looking at the prophages, which is what we call a temperate phages once it's integrated into the genome, we see that 78% of the strains of Baumannii have intact prophages. And when they're found in the genome, they are found to carry really critical genes that totally change the way the bacteria behaves. So these are virulence genes that make these pathogenic. And you can find an average of almost 2 virulence factors in the genome of these strains on prophages. And what that means then is that as these phages excise and leave the cell and infect new strains, and sometimes, they could even shift their host range quite broadly and infect an entirely different class of bacteria, they can bring those genes with them. Not only that, but they can actually take up some of the DNA from their donor cell and move that DNA to a new cell. And so one way to say that is that phages are known to be a mechanism by which there's horizontal gene transfer. And why we care about that, among other reasons, is that, for example, if you take antibiotics yourself and you select for antibiotic-resistant commensal bacteria in your gut, that in itself, if we get rid of the pathogen, is not such a big deal. It doesn't matter too much that antibiotic resistance is now circulating in your microbiome. Now, however, if you were to be infected by a new pathogen, there's the possibility that those circulating antibiotic resistance genes could be moved to the pathogen in situ in your body. And one way by which that happens is through phage. Okay, so these are the reasons that we care a lot about temperate phages. They are critical to shaping bacterial function. They can make pathogens. They can move antibiotics around. But as I said, I'm actually not going to talk anymore about them today, because what I'm most interested in are these guys, the strictly lytic phages, or at least those that primarily use lytic infection. And I am far from the first to think about these. And so this is a paper published by the co-discoverer of lytic bacteriophages, Felix Durrell. And what Felix Durrell did was he was studying cholera patients, and he was collecting their fecal samples and using various filtration methods to filter out things that he knew were smaller than bacteria. At the time, he did not know these were viruses, these phages, but he did know they were capable of replicating and killing bacteria. And you can imagine this discovery is before antibiotics. So the community got incredibly excited about the potential of using phages as therapeutics. We had no way of fighting bacterial infections until then. And so this is a beautiful paper for so many reasons, but every now and again, I go back to it and think how great it would be to be able to publish a paper with only 5 data points. But nonetheless, this is Felix Dorel, who was studying 5 patients that were sick with cholera, and he's tracking them over time. That's the x-axis here. And he's doing 2 things. He's tracking how many bacteria they are shedding, how many cholera bacteria they're shedding in their feces, and that's this dark line. Actually, in this case, sorry, it's the symptoms. He has another tool where another graph where he's showing densities. So this is saying how sick are these people? And then the dashed line is how many of these bacteriophages he's collecting through time. And 2 of the patients died over the course of the study and 3 recovered. And hopefully you can see that in the 2 on the left here where there was death, the bacteriophage population was either negligible, never observed, or took off a little bit and then crashed out and wasn't able to sustain an active phage infection, if you will. Whereas those that recovered had really appreciable numbers of bacteriophage, of choleraphage. And so he used this to get the research community very excited about the prospect of phage therapy. And indeed, he would collect cocktails of choleraphage and go into villages in India where there were cholera outbreaks and dump vials of phage into the wells, with the idea being that he could cure them. And indeed, they would see remarkable stop to these local epidemics of cholera. Now, it was, again, not a particularly well-designed clinical trial because they did not control for the fact that if some strange guy comes into your village from afar and dumps something in the well, you might be right to go to a neighboring village for your water. And that would also stop a cholera outbreak. So, but these trials have inspired recent trials also in India in heavy cholera epidemic areas that's subsidized by Nestle. And so there's still a lot of interest in the use of choleraphage to treat this disease. So here's a small example of it potentially working, but in this case, of course, phages were not administered to the patients. He was just looking at natural abundances. And so one of the things that we do in my lab is ask, when and how phage therapy can work in a more controlled setting. So I'm going to show you now just 3 of my favorite examples with some data behind them of just to sort of convince you that phage therapy can work. Okay. And so the first one is led by Kathy Hernandez, who was a graduate student in my lab, and an undergraduate, Andrea Salazar. And this is just a pretty simple experiment I'm showing you here, where we work primarily on tomato, although I'll also introduce pear trees in a moment as another model system. But in tomato, which we do in the lab and the growth chambers, we can do these really beautiful seedling assays. And so this is a tomato seedling in the black box in the middle here. Can you see my mouse if I'm sort of swirling it around like this?
Yes, Britt.
Great.
Yes.
Perfect. Okay, then I will use it as a pointer then. So in the middle here, there are tomato seedlings, which we've flood inoculated with a bacterial pathogen called Pseudomonas syringae. And you can see on the left, there's no disease symptoms. That's a nice green seedling. And on the far right, a very heavily diseased seedling. And so we allow the disease to establish over time, and we track symptom development. And this is what you're looking at on the left. So we follow how much disease occurs, 3 days post-inoculation up to a week post-inoculation. And when we only inoculate the bacterium, we see that disease progresses beautifully, as we might expect. But when we add our bacteriophage, so this phage 1 here in this case, we have a whole bank of them in my lab, but this was one known as FRS. We see that the disease symptoms really are they're unnoticeable. They're the same as those that we apply magnesium chloride control to. And on the right, this is just a fancy tool we use, droplet digital PCR, to quantify exactly how much bacterial replication and phage replication is happening. And it's just to convince you again that in the absence of phage, we see a lot of bacterial growth. But when we add the phage, bacterial growth plummets and phage growth increases substantially, which should be reminiscent of Felix Dorel's findings from cholera patients. So this is just one example. I'm going to keep going for a minute to show you a few others. This is now, we have a very active research program that is an undergraduate-led project on fire blight in pear trees. And the coordinator of this project is a fantastic postdoc researcher named Dominique Holtepels, who I'm showing here. And Dominique has a group, this is only 4 I'm showing here who generated this next set of data, but a large and thriving undergraduate research group. And what we do is we walk the streets of Berkeley. You might actually see us sometimes if you see people wandering around sampling pear trees in your front yard. Often we try to stick with the ones on the streets, but that's us. We walk around Berkeley and we sample calorie pear. So this invasive ornamental pear tree that's planted by the city very regularly. You might know it for its beautiful white blossoms that happen in the spring, but you might also know it if you have a tree in your front yard that has these little brown disease clusters all over it. So typically these are spread throughout the canopy and they're localized infections. And we've discovered recently that in Berkeley, these little these clusters of brown leaves are caused by 2 pathogens. And I'm going to introduce both of them momentarily. The first one is blossom blight. And this had not been described in California up until we found it a couple of years ago, but it turns out it's highly prevalent around here on our ornamental pear trees, and it has symptoms very similar to fire blight, which I will show you in a moment. What we as a team do is we go out and we sample the trees in front of your houses, and we crush up the leaves. We isolate bacteria from those leaves. And then we also size filter the leaf juice and isolate any bacteriophages that happen to be in that sample at that time and find the host for them. And so through that process of phage hunting, which I love to do, undergraduates love to do, it's a really great exercise, we've discovered numerous new phages. And this one here we call 16Q. It is a phage, a lytic phage that forms what we call plaques. So if you look on the right here, this is a bacterial lawn where we've taken this bacterium, the Pseudomonas syringae pathogen here, and grown it in soft agar. So you get this beautiful, smooth lawn of bacteria. And when you add the potential bacteriophage on top, any single viral particle that infects will replicate lies, replicate lies, replicate lies, until you see what we call a plaque, which is essentially one viral particle that has expanded into a viral population and killed all of the bacteria in that localized area. So, for example, we managed to isolate this phage 16Q that is capable of infecting Pseudomonas syringae from Berkeley. And then the question is, does it actually stop disease symptoms? And one of the neat things about this system is you can just go and collect fruit, pear fruit, either from the grocery store or from Oxford Tract or Gill Tract, where we have pear trees growing. And you can then take a toothpick or a pipette tip, dip it in the pathogen, and then just poke the pear, literally. And you can do that with different amounts of the pathogen or with the pathogen and its phage, for example. And then you monitor the disease symptoms on the fruit. And so on the left, in the top left here, you can see a sort of sad-looking pear that has 3 big lesions caused by this pathogen. And on the right, a pear that's been poked but hasn't developed as severe symptoms. And then we quantify the area of this lesion, and that's our metric for saying how pathogenic or virulent is this pathogen in the presence and the absence of phage. And hopefully you can see in this figure here, these are 2 different strains of the pathogen, both of which we isolated from different trees in Berkeley. And you can see that in both cases, having 16Q phage there at the time of inoculation, excuse me, reduces the severity of the disease. So in the previous tomato example, I showed you that phage are capable of reducing bacterial densities. Here, I'm showing you that they're also capable of reducing the symptoms. All right, there's 2 examples. Let's move on to the third and my favorite from the lab. And this is a recent collaboration between my lab and Dominique Holtepels, who I've already introduced, and Rosa Rowe, who is a researcher in South Korea at the Rural Development Agency. And it turns out that fire blight is a global problem, but especially in South Korea, it's a hugely problematic emerging disease in South Korea. And Rosa and her team at the Rural Development Agency are tracking and trying to stop the spread of fire blight among the pear in Korea and other stone fruits as well. This pathogen has severe effects on production, and unfortunately, apart from spraying like crazy with copper, which is also what we do here in the US, there is no good treatment for fire blight. So hence the interest in using phage in this agricultural space. And so when Rosa came to work in my lab, she and Dominique and I developed I think can officially say the most beautiful experimental assay I have ever worked on, which is quite literally using blossoms or flowers to do our experiments. So what we do is we go out and we sample pear blossoms right when they're emerging in late April, early March. Late March, early April, rather. And so we collect the flowers and we put them down into into water agar. And they will happily stay like that for approximately a week. And then some of them might already have a pathogen in them, but we try to capture them very early in development where they most often are not already infected. And so that means that we can inoculate them with Erwinia amylavera, which is the causal agent of fire blight. And this is just showing you, we've now experimentally inoculated these flowers with increasing densities of Erwinia amylavera bacteria. And hopefully you don't have to look very hard to see that on the left, beautiful, healthy flowers with nice anthers there. And on the right, you see this sort of goopy black disease. And this is actually a biofilm, a bacterial biofilm caused by Erwinia amylavera as it's infecting the flower. And what will then happen in a real system is the pathogen would attempt to become systemic and move into the stem, and then it becomes a war because the plant is cutting off circulation to that blossom in order to stop the spread of the pathogen as it is moving into the stems. And that is why you see the dead cluster, the brown clusters all over the trees in your front yard. So here we proved to ourselves that this is a really neat system. It actually works. You can see more and less disease depending on what you inoculate. And Rosa had come with a cocktail that she is developing already, and she has a patent on, which is nice. And so what she is doing, she wanted to test whether this actually works. So what we did is we had some flowers that we sampled from 6 different trees here. And this is 3 days after we sample and brought them into the lab. The top is those that we did nothing. We just sprayed buffer on. And they look pretty darn healthy after 3 days. The middle is where we only applied the pathogen, Erwinia amylavera, and you can see here lots of disease developing. Not every flower, so there is some heterogeneity still, but when these flowers are infected, they get pretty goopy. And actually, I wish I could zoom in. I need a fancy zoom in feature, but if you look right where my mouse is here, you can see some of these flowers are actively they're producing this exopolysaccharide, this biofilm matrix, and it's literally oozing out of the flower. And this is one of the ways that the pathogen transmits, both within the tree and among trees. Now you might then look on the bottom, and this is the same exact pathogen densities as the middle, but here we've added the phage cocktail. And you still do occasionally see disease. So it's not a perfect cure, but you can see that the disease is greatly reduced in the presence of the phage. And so Rosa went back to South Korea very happy. This is data that she can now bring to the government to show that it's effective and safe. And one of the other pieces of data I'm not showing you here is that a large part of what we were doing while she was here is figuring out whether the use of phage in this natural community disrupts the other bacterial species that are in the flower and might be important for plant health. So that's work that's ongoing. But just to show you that these are the types of number that Rosa likes to take to the government. But in her case, the disease severity index is far reduced in the presence of her phage cocktail. So at this moment, I'm going to stop for a minute to say that I've shown you 3 examples, 4 if you include Durrell's, that phage therapy really can work. And it can, and it's exciting. And I think it has to be part of our more sustainable future as we break our addiction to antibiotics and chemical treatments, especially in agriculture, right? So human health is a little bit more sensitive. At the moment, phage is really only a last resort for most patients, and for some good reasons, which I'm happy to talk to you all about. But in terms of agricultural settings, we rely far too much on pesticides, and this is one currently available option that we could put into the mix to have a more sustainable agricultural system. So what are the hurdles? Well, first of all, I didn't mention this at the beginning, and I should have. Unlike antibiotics, bacteriophages are highly specific. So if you take an antibiotic, it's going to clear the vast majority of bacteria in your system. That is not true of phage. A phage generally has a very limited host range, and so you have to know what you are trying to prevent or cure. You really have to understand the pathogen you're dealing with and find the right phage for that. And I'm excited to say, actually, that the Department of Energy, the DOE, has just given Lawrence Berkeley National Lab a large-scale grant to create a phage foundry. And my lab is part of that. So we're going to be the plant side of the phage foundry. And this is the goal of the phage foundry is to move from where we currently are, which is you have a patient or a plant that's sick, and you reach out to the phage community and you say, help, does anyone have a phage that infects, this particular strain? Move away from that model and move to one where we can computationally predict which phage or phages would be best for a patient or a particular disease. So that's really exciting, and I'm very proud to be part of that team. But that's still one of our big hurdles, that first one. So then let's say you find the right phage for the right pathogen, you still have a scaling issue. So how do you generate high titer of bacteriophage virus to treat that patient or that plant without including all of the endotoxins? So I've already explained to you that phages are obligate pathogens. They need bacterial hosts in order to replicate. And the problem with that is as cells are being lysed, you're ending up with things like lipopolysaccharides and other PAMPs and MAMPs, these molecules that actually our bodies and plants use to mount an immune response. And so when we're inoculating people and plants with phages, we want to make sure that we're also not deluging the system with all of these bacterial endotoxins for which we would mount defenses and sometimes have toxic shock, for example. So that's a big hurdle. Let's say you make it through that one. If we go out to the field and we spray phage, they don't last very long. They're very UV sensitive. They're very temperature sensitive. They're pretty finicky things generally. So application and survivorship in the environment is quite a big hurdle. The one that I'm currently fighting with is regulatory approval. So, getting patents for these phage cocktails and particular processes is very challenging, especially because unlike antibiotics or a sort of typical clinical medicine that you might discover a molecule and try to turn it into a medicine, phages, the power of them is that they can be adaptive. So what I mean by that is if a particular phage cocktail stops working because a new strain emerges that is resistant, the beautiful thing about phage therapy is we add another phage, right? Or we change the phage, we evolve the phage. We can be highly adaptive and in keeping up with a changing medical environment. But unfortunately, current regulatory practices do not allow for that. So after you've gone through all the trouble of patenting your particular cocktail of phage and showing that it doesn't lead to any problems, if you then need to swap out one of your phages for another, you essentially need to start clinical trials over, which is not feasible. And then the last one, which I will actually present some data on now, is that bacteria can evolve resistance to phage, just like they do to antibiotics. I might stop there for a second and see whether there are, as I move away from just the classic idea of phage therapy to a more evolutionary angle, does anyone have questions or thoughts about phage therapy that I can answer? Oh, I see one actually in the chat, which I might. So there's a question from Roger about the risks associated with phage treatment. And to be honest, I would say they are remarkably few. So the folks that I know who administer phage in human clinical settings, the biggest fear is sepsis. So in particular, by the time you're treating a patient with phage therapy, they are typically dealing with a large scale infection, whether it be a highly infected burn wound or a pathogen in the gut that just won't go away. The bacterial densities are generally very large. And one of the biggest concerns is that if you add phage, which by the way is typically done intravenously into the bloodstream, and that works, which I still find amazing, when the phage make it to the site of the bacterial infection and lyse all of those bacteria, there's lots of reasons to think that the patient might go into toxic shock from all of those endotoxins that are suddenly released. But it just doesn't seem to be happening, and nobody knows why. But we've done it enough now. I mean, there have been enough patients that have gone through phage therapy successfully without any side effects, that it seems like a robust finding. So that was one of the biggest fears, and that doesn't seem to be a valid one. In agricultural settings, we have thought about a lot about risks of environmental application, and generally there's very little and no evidence to support any high risk. Okay, then there is one that is actually worth thinking about, because I've already introduced the idea that phages can move DNA around. And so the one serious concern that we would need to deal with as a community is, if you're adding high titers of additional phages into a system, are you increasing the likelihood that genes are being moved around, especially virulence genes or antibiotic resistance genes? And we don't know yet. The evidence for that is pretty mixed. They can, but they don't seem to very often. So I think it will probably end up being a risk-benefit analysis in that one. Oh, looks like there's one more question in the chat. Yeah, so topical treatments is an exciting area, and people have, I know quite a few companies many years ago that were developing topical solutions for multi-drug resistant Staph aureus, for example. But every one of them that I've known never made it to market because of regulatory hurdles. They ran out of funding before they could meet all of the burdens. But I think it's a great place to be looking. And then, really interesting question here. So do we have a phagome that characterizes us as individuals? Actually, I'm gonna come back to that towards the end, but the answer very much is yes. We each have unique microbiomes, but our viromes are far more unique to us. Yeah, absolutely. And typical genome size of these phages in the phages that we work are, 20 to 50K. So, but there are giant phages up to 500 kilobases, and there's very small phages that we don't understand very much about, called helper phage, that I think can even be I just heard about one that was like 1,000. So hugely variable is the answer to the genome size question. The natural level of phage titer in the environment. I can honestly say people have tried to estimate. So I will say in the human body, the original estimates of bacterial densities was 10 bacteria to every single human cell, and that's now been debunked and revised, and it's now about 1 to 1. So you reckon there's about a 1 to 1 bacterial cell to human cell in our bodies. And I would guess that phage are on the order of 10 times that. But yeah, Curtis Suttle has done some really nice work of trying to estimate phages in the ocean and in the environment. And I should remember the statistics, but it's mind-bogglingly high, well beyond stars in the sky high. Yeah. Okay, and then the last one here is why is the regulatory approval so slow, and what specifically are the concerns? And I think the answer to that is fear of the unknown. And so unlike, let me back up for a moment. One of the reasons that we know so much now about the microbiome is because we have a marker, a 16S region of a bacterial genome the 16S ribosomal RNA, for which we can develop conserved primers and sequence all bacteria on Earth. Viruses have no such conserved features. And so our ability to discover the sort of black box of viral diversity is very low compared to bacteria. So we've had to move to metaviromics, where you just, size separate everything that's about the size of a virus and then sequence everything in that pool. But even then, the vast majority of genes that you would see in your metagenome are just genes of unknown function. And if you're trying to convince a regulatory body that your phage, for which the vast majority of genes are unknown function, and you're now going to put that into a system, I think it's fair that there's some wariness about that. But I don't know. I've never been on any of these panels who are deciding. So I don't know what the actual approval hurdles are, but they're very real. Okay, so let's stick to this last one. Let's move to this last point for a moment. So, because this is actually very much related, one of the worries about phage therapy is that if you're introducing a killing bacteriophage into a system, you have the expectation that bacteria can respond, can evolve resistance to the phage. And indeed, they do. So let's stick with Pseudomonas syringae for a minute here. This is just a really simple experiment we ran many, many years ago now, where we looked at Pseudomonas syringae, 3 different strains, A, B, and C here, and we just tracked their growth over time. And all I want you to take away here is that in the absence of phage, this bacterial strain C grew the fastest to the larger densities, but when you add the phage, that changed. And so this is really kind of setting the stage for the end of this talk, where I'm going to tell you that phages can completely change ecosystem dynamics and who wins in a community. But importantly here, now this bacteria C is being actively killed by the phage. And if you put these bacteria into a test tube with the phage and do what we call experimental evolution, where you co-culture the bacteria and phage over time, what you find is that in our case, within 8 hours, usually, the bacteria have evolved resistance to the phage. And they can do that by many, many different known resistance mechanisms. And there's a sort of joke in the field that a new Nature or Science paper comes out every week with a new phage resistance mechanism discovered. We're not even beginning to saturate our understanding of all of the ways bacteria have evolved to defend themselves against phage, but it's a pretty impressive arsenal. I've summarized the top 3 here in terms of commonality, one of which is that phages have to notice a bacterium and use a particular receptor in order to bind to that cell. And a pretty easy way to evolve resistance is to simply change the morphology or the shape of that receptor so that it's no longer recognizable. And indeed, that surface modification is one of the most common phage resistance mechanisms that we see in the lab. The other one is a little thing called CRISPR-Cas, which you now might have heard of for a very different reason, thanks to Jennifer Doudna and team and Jill Banfield, right? This has opened up genome engineering, but of course it was not evolved for that purpose. It was evolved as a, among other things, phage defense mechanism, whereby the CRISPR-Cas region can pick up little pieces of phage DNA and then block the replication of those phages. And this is kind of the closest thing that bacteria have to an adaptive immune system. And then they can also do all sorts of clever tricks in terms of noticing that they're infected with phage and aborting the cell before the phage is able to package and replicate. But again, this is just a very superficial summary of 3 types. There are many, many more. But the point here is that they do indeed evolve resistance. And so going back to that same study I already introduced, what Derek and I did is we just evolved Pseudomonas syringae in a test tube with either no phage, a single phage, or a cocktail of phages. And this is a sort of punchline of a larger story, but what we found is that the bacteria evolved resistance very rapidly, but when they did so, they paid a cost to resistance. So in other words, when a bacteria was able to resist multiple phages, which is this bottom line here, this strain now can grow in the presence of the phage cocktail, which you could say is scary. But the good news is it doesn't grow as well. So it's not as good of a pathogen. Another way of saying that is if you select for bacterial resistance, you're also attenuating virulence. And so that's can be seen as like kind of a win-win strategy for phage therapy. Just to give you another little example of this. So this is work by a PhD student, Sean Meaden, in my group from many years ago, where we did very similar experiment with Pseudomonas syringae. But this time what we did is we evolved the bacteria in the presence and absence of phage. You can see that this is the ancestral bacterium that grows to some density in the presence of phage. But the resistant strain grows to a much greater density. So this is just saying we did indeed evolve resistance. But now when you grow these things in the absence of phage, in a test tube, we didn't really see an appreciable cost of resistance. But when we grow this pathogen in the tomato plant, indeed, the pathogen that had evolved phage resistance is less nasty of a pathogen. It paid a cost for that resistance. I wonder if I want to make sure that I give myself time. You know what? I'm going to do one thing here. And that is jump to this slide. Does this work? Yes, perfect. Okay. So I just skipped a couple of slides and I'm just going to tell you verbally the punchline of those slides, which is I've shown you a couple of examples that bacteria can evolve resistance in a test tube, but I have spent most of my postdoc years showing that that actually does happen in natural settings. So if you track bacterial communities living on a tree in a forest, you can see this evolution happening in real time. So month to month, bacteria are evolving resistance to their local phage. So this is not just a theoretical or a lab effect. This is a very real one. Okay, so if we accept the fact that bacteria will evolve resistance to phage, then there's something pretty clever we can do. And this idea was, I think, really most elegantly and initially developed by a good friend of mine, Paul Turner, who's at Yale, just an absolutely fantastic scientist. But what Paul and his group started to do is think about not just are they paying a cost for resistance, but does that cost have any other phenotypes that we might be interested in? And one of the phenotypes we might be interested in are antibiotic resistance. So he asked a pretty simple question. As bacteria evolve resistance to phage, is there any chance they're becoming more sensitive to antibiotics? And the answer was yes. So in this case, this is just a small example. He's done this in many different beautiful ways now. But you can take a circulating drug-resistant strain of bacteria, select for phage resistance, and then ask whether there was a pleiotropic cost so that they're now once again antibiotic sensitive. And so here is just two examples where he looked at ciprofloxacin resistance and tetracycline resistance. And on the right here, you're looking at the increased drug sensitivity observed as the bacteria evolved resistance to phage. So relative to the ancestor that was drug resistance, and each of these are a different phage here, PAN, PaO1, et cetera. And for many of these phages, the way that the mechanism by which the bacteria evolve resistance means that it is now sensitive to antibiotics again. And I really think this, to me, is one of the most exciting futures of phage therapy, is not being scared about evolution, but taking evolution as part of the tool and saying, okay, either of two things are going to happen. Either my phage is going to work and have its intended effect, which is just that it kills the bacterium and, done deal, great, success, or bacteria evolves resistance, but in doing so, it has this knock-on effect. For example, now it's sensitive to antibiotics or it's less pathogenic, et cetera. And so, for example, in my lab at the moment, we're looking and finding very similar results in terms of copper resistance for bacteria in agricultural settings. And the same thing seems to hold true. So I really think this is just a really exciting aspect of the future of phage therapy. Okay, so I see the chat window. I'm going to go back a slide just to keep that there. I see the chat window. I'm going to take a couple of questions before we move on to the final part of this, which is I'll go through a little bit more quickly, which is the role of phages in the whole microbiome. Okay, so, oh yes. So there's a question about honey as a wound treatment. And has anyone studied phages in honey? Not that I have ever seen. But my understanding is that the actual mechanism by which the antibacterial properties are pretty well understood and likely not phage dependent. But that's not to say that there aren't also phages in there. Phages particularly like that sort of mucosal structure. And one really exciting thing that we're learning is the lining of our gut, which is a mucosal lining inside of our stomach, that phages actually absolutely thrive in that mucosal lining. And you find phage densities in the mucus to be far higher than you'd expect by chance. And that has even led to a new hypothesis for why that mucus is even there as a barrier, that it's not just because it's sticky and stops pathogens moving through, but it's actually a phage trap. And so as any bacteria that's moving through the gut towards the bloodstream has a high probability of encountering a bacteriophage that could kill it. So anyway, so yeah, I think honey could be an interesting, if nothing else, honey as a, as a sort of mechanism for delivery of phage is a pretty neat idea. And I haven't seen anyone explore it. Okay, any other questions before we get up to the wider community question? Oh, it's like one more there. So has Paul Turner's results been used in sequential clinical treatment of infections? So in terms of sequential clinical treatments, do you mean where people are using the different antibiotics, cycling through antibiotics, or?
Yeah, Britt, I was just thinking if you had a fairly desperate case and so many people are increasingly dying of antibiotic resistant infections, then might you apply phage therapy and then at the appropriate time afterwards apply antibiotics again in hopes that there had been mass evolution?
Yes, exactly. They are very much using it and thinking about using it in that way.
Great.
Yeah,
but it hasn't yet been done.
Yeah. They've done it in mice.
Oh,
have they done it in full? I know that Paul is running clinical trials, and I don't know the current status of them, but in mice it works beautifully.
Wow. Thanks.
Yep. And the other thing to say, the other space where I would really watch if you're interested in this is in cystic fibrosis. So cystic fibrosis is a case where many different opportunistic bacterial pathogens end up sadly ending the life of many of the patients who, it's a horrible lung disease. But cystic fibrosis is currently treated with, large and ongoing courses of antibiotics. And so antibiotic resistance in cystic fibrosis is very high. And people have been developing phages that are in nebulizers and inhalers that can get into the lung and seem to really work incredibly well in that space. So in terms of where phage therapy is currently being used and where it's most likely to become approved as a usage, I would say that's right up there. Okay, so what time shall I do this in 7 minutes for those in charge? Is that the goal here? I totally can.
Camille, what do you have a recommendation? I think you probably have the audience enthralled, Britt, so I just do make your choice.
Yeah, Britt, it's fine however you want to handle it. We just try and end around 3, but since you've been taking questions during your talk, we don't have to hold that whole time for Q&A.
Okay, well, I will go fast, but I will try to stay under 10 minutes here. All right, but please keep the questions coming. Okay, so everything I've been telling you about so far is just describing how a phage can be used to control one bacterial population. But this fancy figure here is basically just saying there are many mechanisms by which we actually expect these pairwise interactions to have knock-on consequences to whole community level diversity. And so I'm going to skip the slide just for this one here. So if you were to track your own microbiome over time, so let's say you just use 16S sequencing and you took fecal samples every week and looked at what happened in your microbiome, you would likely see something not unlike this, which is to say that different species become more and less abundant over time. And we're really trying to understand as a field what drives microbiome stability and what drives these temporal changes. And one really understudied possibility is that a lot of it has to do with bacteriophages. And so I'm going to, hope you don't mind, I'm going to use one of my favorite teaching tools here. This is my most favorite way to explain what I mean by this. So frequency-dependent selection, which is what I spent most of my PhD studying in snails and trematodes, is a pretty simple idea that parasites maintain diversity. And this is why. So if you think about the race between high-definition DVD and Blu-ray disc, these two things did the exact same thing. There was no fitness bonus. They had equal fitnesses, right? But what happened is that as one became ever so slightly more popular, it was able to get more titles, and that allowed it to become even more popular and so forth. And so positive frequency dependent selection is the idea that the more abundant you are, the higher your fitness, and that decreases diversity. On the flip side, if you imagine how cool you are as a kid when you come in with the shoes that nobody else in the school has, there is actually a fitness benefit to being rare. There's nothing special about the shoe. You can run exactly the same way in all these shoes, but the rare ones have an advantage, and that increases variation. All right. And so this exact principle can be applied to understanding why we think phages maintain diversity. Because as you have coevolution happening between bacteria in the gut or on your skin or on a plant leaf and bacteriophages, phages are under pressure, under selection pressure, to infect common bacterial types. And as they do, and they kill those common types, they give rare type advantages. And that maintains diversity in theory. And so we set out to test this a couple of years ago. This is work done by Norma Morella, who was a PhD student in the lab. And what she did was, I think, pretty clever. We went out and we sampled tomato plants, the whole community of bacteria and phages that live on leaves. And then we separated those using a series of filtration steps into just the bacterial community, or at least mostly just the bacterial community. We call that phage depleted. That's what stayed on the filter. And then we concentrated the viral-like particles into a potential phage or viral-like particle inoculum. And then in a growth chamber, we sprayed more or less notobiotic plants with either just the bacteria or with the bacteria reunited with the phage from its natural community. And we allowed community assembly to happen. We allowed the microbiome to assemble. And what we found was twofold. First, and we didn't know this was really, we had no idea whether this would be true or not. First of all, these tomato fields that we sampled had a lot of phage. So that was a relief. We didn't add any known phage here. We just sampled what was already there. And when you inoculate a tomato with just the bacteria, you see that there's a lot of bacteria growing on the leaves. When you add the phage back in, those bacteria are being killed. That's what you're looking at here. So, and this is just one day after inoculation. So this got us really excited. Phages are there and they are killing bacteria. So now we have a fair test of the idea, and we could ask, do phage increase diversity of the microbiome as a whole? And for this, we used 16S sequencing, which is just a nice way of characterizing the diversity of bacterial taxa you have in your sample. And what we were able to show is that the plants that received only the bacteria had lower numbers of species, so lower alpha diversity, than if we added the phage as well. And so just like theory predicted, phages were allowing this rare advantage, killing common types, making space for rare types. And this is something that I'm incredibly excited about. And we have worked with a clinical lab to look, and we find somewhat similar patterns in mice. And I think, you know, it's an open question, but as we move towards a a world where we're very nervous about viruses and, using antivirals, we could actually be having unintended consequences in terms of bacterial diversity and microbiome diversity. Just to update this, because this is a slightly older study, here is some recent work by a graduate student in the lab, Rena Debray, and she did a similar thing, but this time, instead of just asking how much phages increased diversity of the microbiome, she asked whether they actually made the microbiome more or less disease protective, with the idea being that if the microbiome is more diverse, it's less likely to be invaded by a pathogen. And I'll just jump to the punchline here, which is that's exactly what we found. So if you spray these tomato plants just with Pseudomonas syringae, which I've already introduced, you see that the Pseudomonas syringae grows on the leaves and causes disease. If you do that in the presence of a natural microbiome, you get slightly less disease, although in this case, not significantly so. But if you add those bacteriophages in as well, then you get a hugely disease protective microbiome. So this is sort of where we're going and where we're really excited about thinking, what role does this viral diversity play in shaping our microbiome health and function? And I mentioned this already, but we've been working with Henry Lin, who's a clinician in New Mexico, and Derek, who I've already introduced, who started as an undergraduate with me while I was a postdoc and is now a MD PhD student in Henry Lin's lab. He's been helping us do this collaboration where we're using a mouse model where they know that if you put them on a high-fat diet, they have inflammation and decreased microbiome diversity. We've been doing viral fecal transplants, so transplanting the viral-like particles from one mouse into another. And we've been able to show that this reduces intestinal overgrowth of bacteria. So again, I won't spend too much time on the data here because I want to make sure I leave time for questions. I'm going to come back to the question in the chat in just a moment, because I want to wrap up with this sort of big thought experiment. So this works. We've done it a few times. We've done it in plants. We've done it in mice. And this led us to my most recent fun intellectual exercise, which is in ecology, and Mary knows this work well. There's been a longstanding question, which is that, is a healthy ecosystem one that is rich in parasites? And there's a lot of theoretical reasons to think this is true. If you have nice, thriving, diverse host populations, you should also have nice, thriving, diverse parasite populations. And we got really excited about this idea and have spent the better half of the last year looking through data sets and trying to figure out whether we can use viromes, so the phage diversity, going back to the question someone asked right at the beginning, are unique viral signatures in our gut. Can we use those viral signatures to determine whether our microbiome is functioning well, or whether we have what's called a dysbiosis? And dysbiosis in this case could mean you have overgrowth of one pathogenic or opportunistic pathogen. It could mean that you just have a shift in your microbiome that's associated with some negative consequence, like a loss of function of something, or you could have a loss of a specific bacterium that you need, or just an overall microbiome function. But we basically have set out to say, regardless of the type of dysbiosis, can we see that signature in the phage? And the exciting answer is yes. So I'll put the data up here for you to peruse, but basically, we found that even when you're looking across these different disease systems, including like fatty liver disease, all the way through to, malnutrition or antibiotic use, regardless of the type of dysbiosis, you're seeing these really striking shifts in the virome of patients. And this can sometimes mean that the viral community just looks really different than it does in the control population. And other times it means that you have enrichment of a specific virus or a specific phage that wasn't there before. And so as an example of this, Cras-like phages, which are very recently discovered and very widespread phages of the human gut, we found that the 9 studies in which we found this Cras-like phage was found to differ in health and dysbiosis 8 had lower abundance in dysbiosis. So just looking at the presence of crossphage, even if you don't know what someone is sick with, it could give you a nice insight into whether it's a microbiome related disease or a non-microbiome related disease. So we're really excited about this and pushing this forward as well. Okay, so that just brings me to my final slide, which is, I hope I have convinced you by now that the microbiome is more than just bacterial. And thinking about the role that phages play, I think is really just getting off the ground and incredibly exciting. And what we've learned over the last 20 years about microbiomes is they're complex, they're variable. We each have a unique microbiome. They're dynamic, they change across time, and they're critical to our health. And hopefully by listening to this talk just now, I've convinced you that phages affect all of those things. So the next time someone talks to you about, probiotics, I say, probiotics, forget the yogurt, eat phage instead. All right. And with that, I'll go back to questions.
Thank you so much, Britt.
Thank you. Oh yeah, crassphage. It's CRASS. Yeah, they're a very dominant member of the phage community in the human gut. And then I certainly shouldn't end without thanking. I mean, of course, I wish I could still do all this work myself, but I no longer can. And in fact, I don't know if my group would let me back into the lab, but nonetheless, I have a fantastic group of graduate students, postdocs, and undergraduates, and they did all the hard work I've just presented. So, and thank you for inviting me. And I will get back to the questions I skipped. So, but please
absolutely great.
Speak out if you have any as well.
Do you want me to field questions, or are you comfortable just reading them, or which is better?
First of all, let me figure out how to stop sharing. There we go. That way we can actually see each other.
Yeah.
Let's see, where did we drop off?
I think it was starting with Roger Y. Is a question that occurred to me too. Are we protected from having phage figure out how to deal with how to use eukaryotic cells? What restricts them to bacteria?
Such a good question. Okay, so if you would have asked me this question even, well, let's say 6-7 years ago, I'd have said, well, I mean, evolutionary history tells us this, right? Like through the entire eukaryotic diversification, there have never been examples, and there are no signatures to suggest that there has been switching of phage between bacteria and non-bacteria. In fact, even the archaea have a very distinct set of viruses from bacteria. So that's what I would have said. However, it turns out, and this is really hot off the press news, that phages are absolutely found in human cells and plant cells. We've done it ourselves. You can visualize phage getting into our cells and plant cells. Now, they do not replicate and they do not cause lysis or disease, but they're in there through a process called transcytosis. Nobody quite understands it, but they're in there in appreciable numbers. And the question now is kind of why and what impact are they having? And this is something that I've been funded by the Chan Zuckerberg Initiative as the BioHub in San Francisco to start asking, thinking about what sort of information could we as an organism be getting from phage around us? Because I don't believe this happens by accident. And even if it is truly an accident, the transcytosis, it's still information. And evolution loves to optimize information use, right? And so this is something we're actively looking at. Again, though, they're not infecting us. They're not infecting human cells or plant cells, but they are interacting with them. And it's pretty cool. And we're trying to figure out why.
Oh, fascinating.
Yeah.
Harold Ogren asks, are there phage in yogurt?
Almost, well, in the yogurt you eat, probably. In yogurt factory, the sort of vats where yogurt is being made, hopefully not. And the dairy industry works very hard to make sure of that. And in fact, CRISPR-Cas, the discovery of CRISPR-Cas as a defense mechanism, which predates its discovery as genome editing tool, was led by the dairy industry trying to find defense mechanisms that they could use to engineer their strains to make them more resistant to phage. Because when a huge vat of culture gets infiltrated by phage, it can take out the entire yogurt production facility, and they need to close the whole factory, do huge sanitation checks and everything. So no is the short answer. Not on purpose. But by the time it makes it to you, maybe.
So Irene Kuhn asks, I wonder if the 30 vegetable a day plan of Dr. Charles Lowry, neuroscientist focused on gut-brain health symbiosis, is partly attributable to the diversity of microbes carried into our guts by drinking this blend of 30 raw vegetables.
Yeah, it's an interesting question. I mean, one of the problems with probiotics is that most of them don't survive into the gut anyway. So, our digestive system is pretty good at killing any bacteria that we eat before they make it into our intestines. So I would be very surprised, but what is certainly happening there is, increased fiber intake is one of the, actually maybe even the only irrefutable fact in microbiome research. So if you eat more fiber, you have a more diverse microbiome. But that isn't, as far as we know, that is not because of the ingestion. It's simply because it's a prebiotic. So it is a complex carbohydrate, hard thing to digest, and it requires a sort of diverse community to do so.
That makes sense.
Interesting idea, though. I don't know. But the phages on those 30 different vegetables, that's another question. Maybe.
Yeah, if I've or Britt have missed any questions, please just speak up.
Okay,
mute yourself and speak up.
Actually, there was just one that was a direct message to me, so maybe I'll just
great
focus on that one, because it's an important one about whether the Russians were, the idea that Russians have been using bacteriophages for many years, and the answer to that is absolutely. Russia and Georgia in particular have had phage clinics since the discovery in the 20s, and they've been ongoing, and they still exist. And this little packet here, where's my camera? This is this is a little packet, a vial that I got. I was a guest at the Eliava Institute many years ago, which is one of the Georgian centers for phage discovery, and they have a pharmacy where you walk in, you explain your woes, and they prescribe a phage cocktail. So they have been doing it, and they have amazing banks of bacteriophages. The problem is that they haven't been doing it in the way that we would consider like blinded clinical trials or anything. That the evidence that they have collected is very, very limited. So they will tell you that it works, and the evidence that they have tend to be thank you letters from patients who've recovered, which are interesting, but not data that we can use.
Right. I want to get to Steve's question, but just on this application of phage clinically, is there a way to stabilize phage so that the phage in your little vial are still viable?
Yes. And the main thing is protecting them from fluctuations in temperature and protection from UV.
And that's all you need then.
That's really all you need. So I have my original phages that a company shipped to me in Oxford, 13 years ago now. I have the original vials. They've traveled across the ocean with me multiple times, and they're still viable.
Great.
So if you protect them, they're pretty stable. Milk apparently also is a fantastic. Some farmers who apply phage therapy actually do it in a milk.
You can see why it kind of coats it, and that's very
exactly.
neat. Steve, maybe you should just speak up and to Britt and ask your question. I feel awkward reading you. Is he there?
Oh,
Steve's asking, to what extent does the presence of thick biofilms prevent the invasion of community by phage? Yeah.
This is also a super active area of research, not just because, so biofilms absolutely protect bacterial cells from phage. there's two answers to your question. I think both equally interesting. So one is that some phages specifically seem to be able to degrade biofilm, and you can imagine why there's great interest in that, right? Like biofilm in medicine is a huge problem in terms of pathogens, but also in terms of biofilms on medical equipments causing huge problems. So having phage that degrade biofilms is really exciting. But the other thing that's neat about biofilms for me as an evolutionary biologist is that spatial structure completely changes the predicted outcome. So what we've done some work, both modeling work and some empirical work, showing that if you have a biofilm, you have phage, infecting and killing the local bacteria, but you create these sort of refuge spaces. And what that means is that you can be a sensitive bacterium in a biofilm and never even experience phage selection. And that means that you really slow the evolution of bacterial resistance in biofilms. So all bets, in terms of translating what we think we know from a test tube into the real world, that's one of the big questions, since biofilms are a very common bacterial growth strategy.
I was just kind of wondering, the cells within a thick biofilm are very physiologically different. Many of the interior ones are inactive, and whether they would be immune to infection.
Yeah, absolutely. I mean, again, just thinking about where we know the most is in rapidly growing bacterial populations in a test tube. And like what in the real world looks like that, grows like that, very, very little. So I think those are absolutely critical questions. The good news, I suppose, is that if you think about many of the pathogens that we're treating with phage therapy, they probably are the closest to growing like that, right? That there's a very active infection and rapid growth. And interestingly, I don't know if this is still true. Steve, you might actually know, or someone else in the audience might know better than I, but tuberculosis is a very slow growing pathogen. And when I was in the Eliava Institute, tuberculosis is a huge problem in Georgia, and they had been hunting for phages of tuberculosis for decades and hadn't been able to find any, which I always found really strange. But maybe it is this slow growing life history that means that phages don't actually do very well.
And they're sequestered within the body in these tubercules.
Yeah,
Right.
exactly. Very hard to access, slow growing. Those are not going to be the ideal hosts of a phage.
So the human host is working against itself in terms of making it at least uninvasible.
Yeah. Yep.
I have two questions. I don't want to speak over anybody else's questions, but if we have time, first, is there more chance for the kinds of genetic recycling or reshuffling that would lead to horizontal gene transfer if you're in a lysogenic situation where the phage is hanging out in the same cell for a while than in a lytic situation?
Yes. Yeah, absolutely. And that's why, so the best practice at the moment when people are developing phage therapy is people tend to not choose temperate phage. So if a phage has an integrase gene, suggesting it can get into the genome, usually those wouldn't be included in a phage therapy cocktail for that very reason, because the prophages, the temperate phage are the ones that are classically thought to be more likely. That said, anything that is killing large populations of bacteria and releasing all of the DNA into the environment, that then means that any competent bacterial cells nearby are more likely to take up that free DNA. So lytic phages probably do accelerate horizontal gene transfer, but temperate phages are certainly the ones that have known mechanisms. They actively do that, right?
I see. Yeah. One other quick question, going back to your initial Petri dish with the plaques in it.
Yeah.
So do those plaques just grow out until there are no bacterial lawns left?
Oh,
or if
great question.
If they don't, what stops them?
The bacterial stop. So once the bacteria stop replicating, the phage stop replicating.
Oh. Well, do the bacteria that are touching the perimeter of the plaque get a signal to stop replicating?
Oh, interesting question.
Quorum sensing something or other?
Anyone's ever looked at that directly. And in fact, that would be really hard to do normally, but now that we're in the world of single cell transcriptomics, you probably could.
Hmm.
I haven't seen anyone do it though. It's a really cool question. Rotem Sorek, who I think is, if he wasn't already here, I know I was going to miss his talk because I'm away, but now I can't remember which trip it was. But Rotem Sorek is coming to give a talk, and he has been pushing forward these amazing well, basically discoveries of how bacteria eavesdrop on phage signals, but also how phages eavesdrop on bacterial signals. So it turns out that
mind-blowing.
It's mind-blowing, but he basically recently showed that that decision about whether to be in the lytic cycle or enter the lysogenic cycle, we've always thought of it as just a probability, but it turns out that phages are actually using molecules in the environment to eavesdrop on how many available bacteria there are, and using that as information to whether or not to enter the genome, which is just mind-blowing.
50K base pairs. How do they do that?
Yeah. Yep. But I really like the idea of looking at the edge of a plaque. I don't know.
Maybe,
And then Irene just followed up on whether that could be due to evolution of resistance. And the answer is yes, but probably then you would see something that resembled like a wavy edge or an edge, something that wasn't perfectly circular, because I don't think you'd expect that high level of predictability if it was resistance evolution. Yeah. And
William Johnston, as the type of phage used against specific antibiotic resistance bacteria described in Strathy's book, Perfect Predator, made any progress?
No, not as far as I've known. So people are really excited about the idea of engineering phage and engineering phage host range. But I'm an evolutionary biologist, and I think, yes, you can kind of stack the decks, but how to say this? Phages are the result of all of these evolutionary trade-offs. And so if you're engineering broad host range, chances are there's some sort of big trade-off. And that trade-off could include, for example, slower growth, slower reproduction within the host, et cetera. And so I have yet to see, I don't know. I don't want to overreach here, but I've yet to see phage engineering really do anything that natural phage variation hasn't already done. But I stand to be corrected for sure. Yeah.
Other questions? This is your chance. Wow. Britt, it was absolutely fascinating, and just it seems that the level of fascination goes on infinitely in the worlds that you and Steve and others here are exploring. It's just so amazing what such, quote, simple looking organisms are achieving in their ecology. That's amazing to me.
Yeah, I agree entirely. Yep. And I think the joke in our field is that the good news is that all that bacterial evolution means job security.
So. Yes, I should think.
All right. Thank you all.
Thank you a million
for the opportunity.